So, good morning, everyone. Welcome to this session on bacteria, vaccines, and immunity. My name is Mark Hatherall. I'm a TB vaccine clinical trialist from the University of Cape Town. My colleague Tom Scriba is an immunologist also from University of Cape Town, and we'll be chairing the session today. We have 10 minutes for each of the presentation. We've got, I think, 8 or 9 presentations, so we'll move swiftly through. We hope there will be time for at least 1 or 2 questions after each presentation. So without further ado, we'll ask the first speaker, who is Sophie Rose, to come up.
Good morning, everyone. So I would like to present some of the work that we've been doing at London School of Hygiene and Tropical Medicine on modelling the response to BC vaccination. But before I actually talk about the specific study, I'd like to just put the work in a little bit of a wider context. So current vaccine development is based on purely empirical methods. Vaccines are tested in mice and then scaled up to humans. And there is some quantitative analysis involved, but we believe that it's not enough. In contrast to this, mathematical modelling approaches, namely pharmacometrics, is used extensively to guide drug development in, for example, trial design or dosing regimens. But vaccine development does not benefit from the same level of quantitative analysis. With new detailed data becoming available on vaccine immunogenicity and efficacy, we believe that there are some exciting opportunities to apply quantitative methods in vaccine development. So I'd like to present an example of how we've been working on this at the school. So we applied mathematical modelling techniques to investigate the human immune response to BCG vaccination, as we think a better understanding of the variation response is required in order to develop new TB vaccines. We have 2 aims of this work. The first one is to develop a mechanistic T-cell model of the T-cell dynamics following BCG vaccination. And then we take this model and we calibrate it to immune response data following BCG. The second aim is then to establish if there are the variation in the responses can be attributed to population covariates. So firstly, I'd just like to show you the data that we used. This is data accumulated from the BCG control arms from MVA phase 1 clinical trials. In this study, the BCG was given at time 0. The number of interferon gamma secreting CD4 T cells was measured using the ELISpot assay over time. As you can see, we've got 4 time points. There are 55 participants altogether, HIV negative, non-latently infected, and UK-based participants. So the population covariates of this group were BCG status, which fell into 2 categories, either BCG vaccinated before vaccination in the study itself, or BCG naive before vaccination in the study. This covariate was then split down further to time since BCG vaccination. So that would be never for those who never received BCG before vaccination in the study, 1 to 9, 10 to 20, and 20 plus years before enrollment into the trial. There was also gender and monocyte to lymphocyte cell ratio, which was measured at enrollment. So that's the data, and this is the model that we would like to fit to the data. So the model is a compartmental mathematical model. It's comprised of 2 cell types, transitional effector memory cell type and resting central memory cells. So very briefly, the cells are recruited into transitional effector memory compartment at a rate delta, which I'll come back to in a minute. These cells either then die or they will transition to central memory type. We have assigned a proportion to the cells that die, which was 90%. The remainder go into the central memory compartment and they stay there for the rest of the time. So as we know, T cells are not the stimulation of T cells doesn't happen immediately after you vaccinate. There is a slight delay, and you can think of this as innate cell responses have to happen before you can see T cells in the blood. So to account for this, our recruitment rate of transitional effector memory cells, delta, which you can see on the left-hand side of the diagram, is actually modelled using a curve, a non-linear curve, which we use a gamma PDF function to model. So when we actually fit this to the data, we believe that all cells in the system are interferon gamma producing. So to calibrate to the early spot data, we use total predicted cells. So that's the transitional effector memory cells plus central memory cells at any time point. To actually calibrate to the data, we use nonlinear mixed effects modelling techniques. These techniques will estimate population mean parameters for the model, but then they will also take into account the variation due to individual responses from the population, and they will and it will assign parameter variation according to this. So the first result I'd like to share with you is just simply, is the model a good representation of the BCG immune response? So if you look at the top figure, the points represent the mean population ELI spot data. The black line shows the model predicted total ELI spot over time. And this is the sum of the blue line, which is the transitional effector memory cells, and the orange line, which is the memory cells. So if you look at the bottom figure, this is now the same model prediction, but taking into account the individuals in the population. The dark blue points represent all immune responses, and the pink bands represent the 5 percentiles from 5 to 95% of model predictions. So as you can see, the model with the variation in parameters covers the data adequately. So the next result is to look into the population and see if there is a variation in response due to population covariates. So there were significant differences in the estimated model parameter values across the population for the BCG status covariate. So this was the covariate that says either you're BCG naive at enrollment to the study or you're BCG vaccinated at enrollment to the study. There were no significant differences in parameters across the population for the covariates gender, ML ratio, or for BCG history. So if you look at the figures, the column on the left is for the BCG naive at enrollment. The top figure is, again, the mean population data for this subgroup. The right-hand column is for the BCG vaccinated at enrollment, and the top figure is for the mean population data. The bottom two figures are taking into account the variation in these subpopulations. And as you can see, for the BCG vaccinated at enrollment, there is a lot more variation in the population, and the model has captured this. So, as a summary, we aim to use more quantitative methods in vaccine development. This particular study shows that previous BCG vaccination is associated with durable interferon gamma responses. Mathematical modelling can provide mechanistic insight into vaccine immune response dynamics. It can quantify these dynamics, and alongside the statistical framework, we can account for population variation in response, and we can also compare immune response across groups. Mathematical modelling can be a vital tool to accelerate vaccine development, and especially used for translational vaccine studies. If I can really quickly just summarise some of our future work. So we would like to rerun this analysis on non-UK human participants if data was available for other geographical locations, for example, maybe an African cohort. We would also like to translate these immune response dynamics to preclinical data. For example, we have some data on macaque immune responses, whereby macaques are split into 4 groups based on their country of origin. We're working on using the model to fit to this data and then analyse which group of macaque best represents responses from the BCG naive and BCG revaccinated participants. if there were data available on, say, TNF readout, we would like to develop a network model, and we can use the modelling to explore vaccine regimen, more doses and timing of doses. But there's still a lot of work to do, but this is a novel method for characterising and quantifying the immune response dynamics to vaccination. So I'd like to thank my co-authors and Helen McShane at the Jenner Institute, who developed the generated data, a colleague at INSERM who helped with the modelling, and this project is funded by ERAS. Thank you.
Thank you.
Thanks very much. Very clear talk, and nice to see a mathematical model that I could understand. At least I think I could understand it. If you have stored PBMCs from the subsequent time points, you could probably test the accuracy of the model, couldn't you, for the central memory cells by flow cytometry to look at, or by cultured ELISpot? Are you planning to do that?
Yeah, that's one of our aims. So we don't, I don't have that data currently, but aside from this work, we've been doing a lot of work in mice and using the same model, and we have frozen cells from that we plan on running flow cytometry on, so we can actually characterise how many memory cells are, how many effective cells are. So it's a plan for the future.
Because it would be great to have a model that's, that's validated as well.
Exactly, and that's
the field, yeah.
Exactly, and that is our plan. I mean, the model is kept quite simple purely because we have only the total cell counts and 4 time points, so we keep it very, very simple. But if we had cell characterizations, then we could be a lot more confident about what our model is predicting. Thank you.
Thank you, Sophie. Sorry? No, you have to keep it clear.
Okay, I'll give you a very quick question. So on your last data slide, could you go to your last data slide? The BCG revaccination model, oh, you can't.
No, sorry.
Okay. But wouldn't you expect that your central memory response is the one that accounts for the higher baseline and people who've been vaccinated before, whereas you started again with a 0 memory response?
So this is an assumption of the model okay, no.
It's fine.
So an assumption of the model is that if you have an existing memory response, when you're vaccinated, this sort of immediately turns into an effector memory response, which is one of the assumptions that we're trying to work around when we develop a new revaccination model. But that's a really valid point, and we're going to be working that into the new version model.
Thank you. So the next speaker is Ed Nardell, and he will be asking the question, can BCG prevent M. tuberculosis infection in guinea pigs exposed to human cases?
Thanks, Mark, and pleasure to be here. This is a report on a study funded by the Bill and Melinda Gates Foundation, and this was essentially following the disappointing results of the Oxford vaccine trial, and an attempt to sort of redefine animal models, because previous animal models had suggested that, in fact, the Oxford vaccine would likely work. The other reason for this study was an interest in alternative strategies, one being that vaccines might actually prevent infection, and to show the interest in that, in November of 2 years ago, a symposium was held, or workshop was held at NIH, and the rationale is that preventing infection is probably better than preventing disease, and less expensive trials because there are more events, perhaps, and may discover novel vaccine mechanisms. On the other hand, as there was some concern ultimately down the road in preliminary discussions with FDA, questioning whether, in fact, this vaccine would be more likely effective in the 90% of individuals who actually don't go on to disease anyway, and also we don't really have a good gold standard for infection, and that's a problem as well. So just to point out where, in fact, this is the pathogenesis of TB in low-burden settings, and it's sort of the one we think of more often than in high-burden settings, but suggesting that there's a primary event with primary tuberculosis, clearing, and hematogenous spread. And what we were always taught was that BCG prevented hematogenous spread, particularly TB meningitis, but that it didn't prevent infection. In part, we had no way to measure the prevention of infection. But subsequently, there's been a number of observations, and David Lavani, who's in the room, was responsible for several of these, and the review that's listed here at the end, suggesting that, in fact, with the advent of IGRAs, that in various settings, that there were fewer IGRA positivity and exposure settings among those who had had BCG vaccination, and including in adults in a Taiwan prison. So there was some suggestion that maybe BCG does prevent infection after all. Now, the problem is with the laboratory models we have up to date, when the guinea pig is used, very often 15 to 30 colony-forming units are used, and with various strains of laboratory tuberculosis, invariably these animals all get infected by intent, and they all die. Whereas with the model that I'm going to show you, we were sacrificing animals long after this and showed no evidence of progression of disease in many cases by that time. So a very different situation. And so what you were left with the guinea pig model and many other models is looking at vaccinated, not vaccinated in terms of colony-forming units. So could you reduce colony-forming units, but really no possibility of looking at preventing infection. Now, so we exposed guinea pigs in this facility in Mpumalanga, South Africa. It's an MDR-TB hospital. It's now become more an XDR-TB hospital. We have 6 patients in rooms, and we send all the air to 2 exposure chambers where we have up to 360 guinea pigs. And for this study, we used all 360 guinea pigs. Our first study also had 360 guinea pigs. We showed very nice skin test reactivity in the first, after a month of exposure, and by the end of the study, 75% of the guinea pigs were infected by virtue of a tuberculin skin test reaction of greater than 6 millimeters. What's interesting is that we actually saw, and these are individual guinea pig responses, very clear evidence of transient responses, presumably transient infection. All of this could be suppressed by irradiating the air. So this is not a response to dead organisms, but up, 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 down, and this happened in actually quite a few animals. And then even reinfection was very clearly seen, or because exposure was continuous. We didn't remove the animals after their initial reaction, simply because we had no place to put MDR-infected guinea pigs. But it turned out to be quite an interesting happenstance. So overall, in that particular study, we had infection in 75% of animals, but reversion in 15%, reinfection in 9%, and disease in about 15%, not unlike human rates of disease. So this stimulated the Gates Foundation, and in collaboration with ARIS, to engage Diane Ordway and Ian Orm's group in Colorado State to take a much more intensive look at the immunology to vaccinate a percentage of guinea pigs. And Dominic Gladdy at ARIS was intending to try and develop a IGRA test. And that actually didn't happen. I mean, they were unsuccessful, I should say. And I should mention my South African colleagues at the MRC and at the University of Pretoria. Anton Stoltz was the on-site PI, our chief technician at Cubus Venter. There are many differences between these 2 models, as I've mentioned. We were dealing with pathogen, with natural infection. The guinea pigs available in South Africa are not pathogen-free. So when we know that gut flora influence immune response. We're using cultured bacteria in laboratory studies, and here we're using host-generated, low doses of host-generated bacteria, unpredictable in terms of number and unpredictable in terms of strain. So ultimately, I'll just mention that the Gates Foundation went on to decide that they'd rather have some sort of hybrid, and so they're using a primate to guinea pig model where they can choose a strain, and it's still not the number, I suspect, but no reinfection in this laboratory model, here reinfection, and we had some non-persistent infection. Were they infected? How do you define infection if animals are transiently responding and then don't? And we have evidence from that first study that even with immunosuppression, they did not go on significantly to disease. We think they cleared the infection. Tools for guinea pigs are not many, but Diane Ordway and Ann Rawkins at Porton Down have quite a few. This was the plan. We had 4 groups of animals. We first started off with drug-susceptible TB because that's what actually the Gates Foundation wanted us to do. They're not developing vaccines against MDR and XDR strains, so we never did that before, and it turned out to be quite a challenge to get people from the clinic to the air facility for 48 hours and back to clinic so that we didn't delay their treatment. And we did it, but, and you'll see that it had some impact. And then after a period of time, we switched to our older pattern of using MDR and XDR patients, and that has consequences as well. The methods that were used by Diane, and she should be presenting this immunology, was bacteriology, flow cytometry for cell simulation studies, cytokine responses, and histology. So the full range of things. 90 animals were BCG vaccinated, 90 animals were sham vaccinated, and part of the naive animals were to develop this IGRA test, which was unsuccessful. In getting people from the clinic to the air facility for 48 hours who were all gene expert positive, we found ourselves, especially in a high HIV setting, with patients with very little disease. They were quite early in their course. This was a first presentation, as opposed to the chronic failure patients that we had been treating who have XDR TB and MDR TB, who had much more advanced disease, but strains that may be less pathogenic. So in terms of the naive guinea pigs versus the laboratory ones, Diane noted a lot of differences in the cytokine pattern. The pathogen-free animals actually had much more in terms of IL-17, pro-inflammatory, and more in terms of suppressive IL-10 compared to our naive, our naive but pathogen-exposed animals. This is the bottom line of the results, and yet this is not the results that I was asked to present, because we were talking about does BCG prevent infection, and what this is about is colony-forming units as determining disease. So this is a bacteriology result, and it's not a infection result. And as a result, so in our using the TB drug sensitive, we did in fact have 7 infections only, very low rates of infection for us, and none in the BCG vaccinated, but not many in the one in the sham vaccinated as well. So very small numbers and hardly convincing, but nonetheless no TB noted by bacteriology in the BCG vaccinated. When we switched to MDR, we had more infection, but clearly no evidence of any protection there. But again, we're talking about disease here, not infection, and that is, to be honest, the answer in terms of whether we prevent infection is not. We haven't really concluded. As you'll see, there's an awful lot of immunology to decipher. So, in terms of cytokine responses, you see some here. This is log over baseline. And it suggests that there were no disease cases, and so we had similar rates. This is bacteriology, sorry, and let me just move along here. And these are individual animals. The animals broke out, as we saw with the skin test, into high responders and low responders. And you'll see that it looks like some progressed with a higher disease burden and some did not. And same with the MDR cases. And what's different about the MDR cases is much more of this transient infection. That may reflect low virulence on the part of the organisms. We have a great deal of trouble culturing drug-resistant strains from the guinea pig. We saw pathology, and one of the concluding things here is that with ongoing transmission, Diane's hypothesis here, and I agree with her, is that there may be some immune exhaustion going on with complete recurrent reinfection that then allows some animals to go on to progress to disease and others not. And I think the role of reinfection in really high burden settings is not fully appreciated or understood yet, and it's something that needs to be pursued. And I think we're just about done. So protection seemed to be incomplete in this model, but again, we haven't concluded in terms of infection. The difficulty of defining infection when you have so many measures, none of which are gold standard, is a challenge, especially since transient infection is a part of it. Pathogen virulence is probably a factor, and reinfection is probably a factor. List of the participants, and just to mention that we're actually doing a study in adult travelers to high burden settings of BCG, funded by the President's Initiative on Combat Drug Resistance. We can talk about that afterwards. That is it.
I was just interested in the transient sort of, let's call it changes that you saw. I presume these were cytokine changes or changes in gamma where you saw this change in the guinea pig. What did you find in those animals where you saw this transient response? Did you find pathology?
No pathology, and when we took half of the animals in that first study, immunosuppressed them with corticosteroids, high doses, and could not produce disease. So it appeared that they cleared this infection. There's this idea of now trained immunity, something between innate and I think that's what we're triggering is some initial response that then is successful in clearing a very low dose of infection.
No, I asked this question because, when healthcare workers are screened, or when we don't use IGRIS in South Africa, but where they are used, you often see these transient responses, and you wonder what it means.
Well, I think under low burden settings, it probably has a lot to do with test characteristics. In high burden settings, I think it may actually reflect clearance, reinfection, that kind of thing, especially if the changes are big.
But even with steroids, you couldn't demonstrate.
We couldn't.
Thank you.
Who's going to tell us about lineage 7 of MTB?
Thank you for the introduction. I will follow this outline for my presentation. And I will start my presentation by briefly describing what lineage 7 is. Lineage 7 is a newly discovered MTB lineage in Ethiopia. Phylogenetically, this lineage is deeply rooted and lies somewhere between the ancient lineages of lineage 1, 5, and 6, and modern lineages of 2, 3, and 4. Lineage 7 has recently started to diversify, and in one of our earlier studies, it was shown that lineage 7 constituted the second highest proportion of the collected strains. So what motivated us to do the current proteomic research? Again, in our recent study published on JCM, we showed gross difference between lineage 7 and other lineages. More specifically, the colony weight and colony diameter of lineage 7 strains were significantly lower compared to other lineages. So in this study, we wanted to see what factors may have contributed to slow in vitro growth in lineage 7. This is a proteomic pipeline that we use to analyze the data. First, we subcultured strains from lineage 4 and lineage 7, and then cell lysis and injured trypsin digestion was performed using the standard procedure. And then the peptides were injected into the mass spectrometry for peptide characterization and quantification. And finally, protein identification and, again, quantification was performed by MaxQuant and Persis software, respectively. So what did we find? This Venn diagram shows the protein overlap and the total proteins identified. In total, we identified around 2,867 proteins, covering 71% of the total proteome of MTB, which in fact is higher compared to several studies conducted so far. And the second, this map shows the differentially regulated proteins. We found around 125 differentially expressed proteins out of the total. And this heat map shows the down-regulated and up-regulated proteins in relation to the strains. We found a total of 64 down-regulated and 61 up-regulated proteins. These were significantly different proteins. The other interesting finding is that we found downregulation. Lineage 7 showed downregulation of phosphate binding proteins, which are involved in phosphate uptake. As you all know, phosphate is essential component of DNA, RNA, ATP, and it's involved in several metabolic processes. So the downregulation of these phosphate binding proteins in lineage 7 may affect the phosphate-specific transport system activity, and that will lead to phosphate depletion to lineage 7. Earlier studies have shown that MTB with disruption in genes encoding the PST system exhibited decreased virulence, and a PST knockout mutant showed attenuated growth within the macrophage. So the downregulation of proteins involved in phosphate uptake may affect lineage 7 growth. The second important finding is that lineage 7 exhibited the downregulation of proteins involved in E6 secretion system. As you know, the E6 secretion system is essential for mycobacterium-mediated iron acquisition in mycobacteria. So the downregulation of all these proteins, shaded in green, may lead to phosphate starvation iron starvation in MTB lineage 7. This cytoscape analysis shows how the downregulated proteins interact with each other. Earlier studies have shown that the E6 secretion system is essential for in vitro growth in MTB, and the lack of siderophore export system significantly decreased the number of MTB cells in infected mice. So the downregulation of almost all components of the E6-3 secretion system may affect lineage 7 growth. Apart from this, lineage 7 exhibited downregulation of several proteins involved in counteracting the oxidative and nitrosative stress exposures, which may affect also its growth. In conclusion, this study generated a novel knowledge regarding the differentially expressed proteomic profile of MTB lineage 7 versus lineage 4, and the downregulation of several component proteins of the E6-3 secretion system and the PST system may lead to inorganic phosphate depletion and iron starvation in lineage 7, and that may lead to slow growth in lineage 7. Therefore, the slow in vitro growth that was observed in our earlier study may be considered as possible phenotypic consequence of lineage 7. However, we recommend further proteomic study to fully understand the phenotypic consequence of the differentially expressed differential expression profile of the lineage 7. Finally, I would like to thank our group members in Norway and institutions who have participated in this work, and thank you all for your attention.
Thank you very much for that clear presentation. Maybe I can start. How many strains from lineage 7 and lineage 4 did you compare?
2 strains from lineage 7 and 1 from lineage 4.
And so did you again see that lineage 7 grew slower? I mean, how did you standardize?
It still grows slowly.
Okay. And so how did you standardize your bacterial preparation so that you can compare the protein fractions?
we did the same procedure for all the strains actually. So we took all precautions so that everything is okay for the strains to grow actually.
Yeah. So, how do you know anything about how this strain performs clinically compared to other strains? Is it comparable to the low in vitro activity?
Yeah, this is an excellent question. And we did a study 2 years ago, and we compared, for example, diagnostic delay between strains, between patients infected with lineage 7 compared to other lineages. And we found an interesting finding that patients infected with lineage 7 were significantly delayed in seeking health care compared to patients infected with other lineages. So we have to do now studies on immunologic profile of these patients. That's in our plan. So there is clinical difference among.
But any differences in pulmonary versus extrapulmonary, or?
Yeah, well, in our earlier study, we took only pulmonary TB patients, not extrapulmonary TB patients.
Yeah. Okay, thank you.
Yeah.
Thank you.
Yeah. Okay.
Okay, our final talk will be from Yan Huang Wang, I think, or yeah, from Taiwan, and he will tell us about the inflammasomal response of the ESET6 stimulation.
Thank the organizing committee for giving me an opportunity to share our preliminary data on the difference of inflammasomal response between active TB and latent infections. I'm a pulmonologist in National Taiwan University Hospitals. Okay. Why conduct this study? We all know that the predicting active TB from latent infection is difficult by current diagnostic test, and it is the major limitation in the intervention of latent TB infections. Previous study demonstrated that Mycobacterium tuberculosis is capable for inflammasomal activations and is very critical in host immunity and host infection control for tuberculosis infections. And however, currently the human data are lacking. Therefore, we conducted these studies. We hypothesized that inflammasomal response of peripheral blood monocyte-derived macrophage after TB antigen stimulation can differentiate active TB from latent infections. In this study, we first collect blood samples from patients with cultural confirmed pulmonary tuberculosis and close contacts who were quantiferon positive. And after blood sampling, the peripheral blood mononuclear cell were immediately isolated by FICO. And after isolation, the human CD14 positive monocyte were isolated by the MAX system. This is a commercialized CD14 positive selection systems. And the isolated monocyte will be cultured for 5 days and allows to differentiate into macrophage in the presence of macrophage colony stimulating factors. And this is a picture This is a picture of culture plate during the differentiation process. And at day 4, we change the medians. And this is a demonstration of flow cytometry at day 6. And by comparing the surface marker stain and isotype stains, you can see that the PB monocyte-derived macrophage has already lost their CD14 surface markers, but they have strong expression of another 2 surface marker of macrophage, that is CD86 and CD206. And they was weakly positive for CD68. And the derived macrophage was stimulated in 6 antigen preparation. And in the preparation, the lipopolysaccharide were used as positive control. And the experimental group consists of 2 levels of ESET6 and 2 levels of protein derivative, purified protein derivatives. And there were 2 durations of stimulation. The 6-hour stimulation is for subsequent assay of gene expressions by Q-PCR. And the primer used in the Q-PCR is listed here, and GAPDH is a housekeeping gene. And another culture duration is 24 hours, and this is for the subsequent cytokine assays by ELISA. But I'm sorry, we haven't done the cytokine assay yet. And these slides demonstrate the results of qPCR in the contact with latent infections. By comparing with GAPDH, you can see that the tumor necrosis factor alpha, the gene expression of tumor necrosis factor alpha and interleukin-1 beta is increased and in a dose response pattern. However, there is no significant change in the gene expression of other 2 genes. And this study is currently ongoing, and we have analyzed 7 TB cases, the qPCR results of 7 TB cases and 19 close contacts with latent infections. And the blue bars represent TB cases, and the orange ones represent contacts. And this is the stimulation of this is stimulated by low-level ESET6 and low-level and high-level ESET6 and low-level PPD and high-level PPD, and this is a positive controls. And the y-axis is the log transformation of fold of expressions. And this picture shows that the gene expression of interleukin-1 beta after ESET6 stimulation is a significant increase in contact compared with TB cases. And in contrast, the gene expression of tumor necrosis alpha after antigen stimulation is significantly increased in TB cases. And so forget the first conclusion, I think. The study, we have so many problems during in the laboratory procedures, but based on our preliminary data, we think that our hypothesis is correct, and the inflammasome response is different between cases, between TB cases and the context of latent infections. And however, I think we need to further optimize our laboratory procedure and do further cytokines assays. And of course, we need more TB cases and contact to compare, to confirm our study results. And I thank all who sponsor and who participate in this study, and thank you for your attention.
Thank you very much. Okay, I'll start again. So what is your hypothesis around ESET6 stimulation of the macrophage? How do you think the macrophage can recognize ESET6?
Yeah, we were not sure before our design our studies, but according to the previous studies, mycobacterium tuberculosis is capable of inflammasomal activations. So in this study, we not only use ESET6, the specific antigen, we also use purified protein derivatives. But we see that for antigen mixture of tuberculosis and specific antigen, they all have inflammasomal response. So I think this probably supports our hypothesis.
Mm-hmm. Hazel?
I think it's a very interesting approach to look at the inflammasome. In these contacts, though, do they get prophylaxis, or do you get a chance to follow up to see what happens? Did any of them develop TB?
Yeah, thank you. This is a very important question, and we collect the blood sample before. Actually, this is performed in a prospective studies and to follow up the contacts who before receive preventive therapy and then after receive preventive therapies, and these samples were collected before preventive therapies, and so we will collect their blood sample at first month after treatment and 1 year after treatment. But the study is currently ongoing.
Okay, because there are also some data sets now with gene expression, data sets from contacts who did progress to TB. And Tom Scriber, one of our chairs, and Dan Zak have published some of those. So you could look for some of the inflammasome genes, and I don't know whether, Tom, you've looked at those.
So, I mean, so IL-1 and associated gene expression is upregulated as people progress to TB. Great. Well, thank you very much for keeping to time and for the great talks. And this ends the session.